I'm pleased to welcome Claudia Bonfio. I'm sorry, Claudia, we did not receive a bio, so the information I have is very limited, but I believe you're with France's Institute of Supramolecular Science and Engineering. I hope that's correct. And you may want to give a few additional details if you think it's relevant. And we look forward to hearing what you have to say about the emergence of modern cells. Claudia.
Thank you very much. So sorry for the bio sketch and for the few technical issues. As you said, I'm Claudia Bonfio, and I am Italian. I had my PhD, did my PhD in biomolecular sciences in Italy with Sherif Menzi, and there I was working already on origin of life, and I was working with iron sulfur clusters. And then I moved to Cambridge at the MRC Laboratory of Molecular Biology, working with John Sutherland. And there I focused on another building blocks of life, which is lipids, which are lipids. And so at some point I started to think about combining sort of all the building blocks of life together. And so that's the origin of my talk and also the origin of my title. And right now I am a junior group leader in Strasbourg at the Institute for Supramolecular Science and Engineering. And so I'm still focusing on the origin of life from a chemical perspective, a bit at the interface with biology. So if we go to the next slide, and I'll just thank David already from the very beginning because he is going to change all of my slides. So thank you very much for that. So if we think about modern cells, we've heard a bit about modern cells in the previous talk. If we think about modern cells from a chemical perspective, we know that they are sustained by chemical reactions that are very frequently interconnected. And these chemical reactions are directed by nucleic acids, they are powered by proteins, and they are protected by lipid membranes. And so these are the three main ingredients that we can identify, chemical ingredients that we can identify in modern cells. And so if we move to the next slide, it will be, quite easy to notice that when we think about how cells might have emerged on early Earth, we're thinking about primitive cells that were likely relatively similar. They were likely based on the same three main components, primitive versions of nucleic acids, of proteins, and of lipids. What we don't know is how the transition happened, how we go from the simple building blocks of life to a primitive cell, and then how we go from a primitive cell to a modern cell. So what I thought was important, especially because of the multidisciplinarity of this meeting, of this conference, was to think about and discuss together what primitive cell mean and what we mean by primitive cell. So if we go to the next slide, that's exactly the first question that I want to try to answer with you. And when we talk about primitive cells, we have another way of calling exactly the same thing, which is protocell. If we go to the next slide, you'll see that. So what does primitive cell, what does protocell mean? And luckily, someone provided us with a definition. So if we go to the next slide, we can see that the definition that was given for primitive cell or protocell is basically a model that involves a compartment, a self-assembled compartment that is linked to chemical reactions that can take place inside this compartment or around this compartment. And this model aims at explaining how modern cells might have emerged. And so if we go back to this, next. But if we go back to the figure that I showed you before, which is how we depict a primitive cell, then as we just said, a primitive cell is basically a container and then some content inside. And so if we, one of the questions that I started asking myself was, okay, so there's a lot of people that luckily is progressing quite a lot in understanding what the building blocks of life might have been and how they might have emerged on early Earth. What is less explored so far is how they interact with one another. So if we go to the next step, the question basically was, we have the building blocks of life. They might be around or they might be contained inside these primitive compartments. What happens when we have all of them? What happens when we have the compartment and the content in a primitive environment? So if we go to the next slide, you can see that the question I aim to answer was, assuming that we have compartments, and in this case, we are focusing on membranes, and we know we are going to have a talk later that talks about different kinds of compartments. So if we think about compartments as early membranes, how do we know that the chemistry that we want to run inside of these membranes and that we believe is prebiotic chemistry, how do we know if it is compatible at all? So if we go to the next slide, you can see basically you can see the starting point that we decided to explore. We had developed in the lab what we call activation, an example of activation chemistry, which basically means that we developed a chemistry that can be used to activate some of the functional groups of some of the relevant building blocks of life. In this case, we have on the left a nucleotide, and then depicted in drawn in orange, we have the representation of an amino acid with carboxylates. And what we wondered was whether we could develop a chemistry that could activate these functional groups to increase in complexity the building blocks that we had in solution. And what you can see on the right part of the reaction is that we managed to develop an activation chemistry that could form more complex systems. So it could activate both phosphates and carboxylates. And this was not only happening when we used nucleotides or amino acids, but if we move next, we can see that this could also be applied to basically any kind of phosphate group or carboxylate group that we had in our hands. So the idea here was just to find, let's say, a relatively reliable starting point, a relatively reliable chemistry that we had explored in the lab, and combine it with the compartment, trying to see whether this chemistry would work inside compartments. So if you move next, you can see what kind of compartment we are thinking about here. So the compartments that we looked at were made of fatty acids, and these are molecules that were thought to be relatively abundant on early Earth, relatively primitive, and they are very simple molecules that have a hydrophobic component and a hydrophilic carboxylate in the head group, and they are known to self-assemble into lipid membranes. So we had our compartment, we had our chemistry, and what we decided to do was to understand what would happen. So the first experiment that we run in the next slide is exactly the same kind of reaction that I've shown you before. So a nucleotide that has a phosphate group and we exposed it to activation chemistry. And the difference this time is that we exposed it to activation chemistry in the presence of lipid membranes. And what you can see next are the NMR traces of the solution of the reaction that I just discussed with you. On top, you can see the reaction in the absence of compartments, in the absence of the lipid membranes. On the bottom, it's the reaction with the membranes. Now, the outcome of the solution is what we call a cyclic phosphate. And you can see that on top, after 24 hours, we have a complete conversion of the starting material to the final product. You can see on the bottom NMR that the major species is exactly the same. So we still are capable of replicating the same kind of activation chemistry inside lipid membranes. But here we have that we notice that the reaction is slightly less efficient, and you can still see some A3'P, which is the small peak at 2 ppm, which is the starting material. So the reaction does not go perfectly to completion in the same amount of time. But you can also see a new peak. And if you move to the next slide, I'll show you that the peak is basically due to the activation of the components of the lipid membranes, which are carboxylates, as we can see on the bottom, and their activation drove the synthesis of 2' acylated nucleotides. Now, what kind of acylated nucleotide are we talking about here? If you look at the next slide, you can see that the molecule that we have here is, of course, with a nucleotide, but now it has a relatively longer acyl chain, and this acyl chain is hydrophobic. So this molecule, we went from something that is very soluble in water and very hydrophilic to a molecule that is now conjugating a hydrophilic and a hydrophobic part. So what we were interested in looking at was how this molecule would behave in water and how it would behave in the presence of lipid membranes. Because it's already known, if we go to the next slide, that molecules that have this kind of shape, so that have a hydrophilic and a hydrophobic component, can self-assemble into supramolecular structures. We've seen that fatty acids can form lipid membranes, but they can also form micelles, which basically are much smaller supramolecular structures where you have the lipid with the hydrophobic components pointing inside and the hydrophilic group pointing outside. And what we already know from this is that if you mix fatty acid membranes and fatty acid micelles, the membrane grows. So basically, if you have molecules that self-assemble into micelles in solution, they are going to feed into the lipid membrane and make it bigger. So we were curious about whether the molecule that we've just synthesized by exploring prebiotic chemistry, activation chemistry in membranes, could do exactly the same. And what you can see in the next slide is that, yes, it can do exactly the same. So we noticed that by adding this molecule in solution, it could self-assemble into lipid micelles, and then adding this to a solution of lipid membranes, you could observe the increase in the size of the initial lipid membranes. Now, we were curious to know whether this was something that would only happen for this kind of system, or it could be a more broad finding. And so what we decided to do was to change the head group. Instead of using nucleotides, we decided to look at amino acids. And so if we look at the next slide, you can see that we made a system that is very similar. So in this case, we have an amino acid as the head group and still the acyl chain as the hydrophobic component. And you can see that this molecule behaves exactly in the same way. It assembles into micelles, and then feeding into the membrane, it makes it grow. Now, what's interesting about amino acids is that we know amino acids have a lot of different side chains. So we could play quite a lot with the nature of the head group of these conjugates. And the amino acid that we decided to look at here is arginine. Arginine has a positive charge on the side chain. And so here, not only we have a molecule that combines a lipid and an amino acid that can self-assemble into lipid micelles, but also we have a head group that is now positively charged. Now, we know that fatty acids, you can see it from the fact that fatty acids contain a carboxylic acid group that is frequently, at least partially, deprotonated. So you can imagine that the lipid membranes that are made of fatty acids are negatively charged. And so this was quite an interesting finding, because we had positively charged species that were feeding to and localizing on negatively charged lipid membranes. And so what we decided to look at was whether we could exploit it somehow. So if you look at the next slide, you'll see that what we were interested in doing was exploiting the positive charge that was present in these amino acid lipid systems and localize molecules that were already negatively charged, and so they would never localize or interact with the lipid membrane that is negatively charged as well because of repulsive effects. And so what we did was we started from a lipid membrane, and we had in solution some RNA, just because we know that RNA is negatively charged. And we decided to add our positively charged amino acid lipid conjugates. And what we observed was that because of the presence of positive charges on the lipid membrane, RNA was now localized to the membrane. In the next slide, you can see the microscopy images that we collected, and the bottom row is representing what I just described. So we had fed the membrane with positive charges, and now we can see that the RNA that is labeled in green is perfectly localized to the lipid membrane. So if we conclude this first example of combination, let's say, of different building blocks, and we go to the next slide, we can say that this specific kind of prebiotic chemistry that we looked at, that is activation chemistry, can drive not only the same chemistry that we were used to see, but also it can drive the formation of lipid membranes that now are even more advanced. So this seems to suggest that we should actually be looking at the coevolution, the compatibility and the combination of different building blocks of life. And in particular, if we go to the we've seen that in particular, we can make new species that combine some of the building blocks of life that most rarely are explored. And this can drive, as I was saying, the emergence of primitive cells that seem to be much more functionalized than before. Now, if we move to the second story that I wanted to share with you today. So when we depict, as I showed you before, when we draw a primitive cell, we normally draw it like this. We draw it as a series of chemical reactions interconnected, hosted inside the primitive cell. And we normally draw that as if nutrients can be taken in from the outside, and products or byproducts and waste can be released outside. Actually, this is not what we do in the lab. So if you look at the next slide, you can see that what we really do in the lab is we put the nutrients, the substrates of our reactions already inside the primitive cell. And also, no one has really focused that much on getting rid of the waste. So I guess that the question I had was, assuming that we have a very efficient reaction that runs inside our primitive cell, at some point we get to accumulate so much of the product that we have to either use it somehow or get rid of it in some way. So and at the same time, we might not be able to have the nutrients already present, the substrates already present inside the primitive cell. We might need to get them from outside. So what I was interested in was understanding how we can actually mix the content of different primitive cells and have them exchanging their content. So the question in the next slide that I asked myself was, how could primitive cells mix, reshuffle their content? Again, the kind of systems that I was interested in, as you look in the next slide, are compartments made of fatty acids, as I just said before, because they are considered very prebiotically relevant. And the main reason I decided to look into that, let's say the starting point I decided to look at was a paper that was published about 10 years ago. If you go to the next slide, it's a paper that was published by my PhD supervisor, and that would look at the thermal stability of lipid membranes made of fatty acids. And what they saw, if you look at the next slide as well, is that fatty acid-based membranes were thermally unstable. And they could see that playing with the size of the lipid that was used or playing with the size of the content, they could modulate the release or the uptake, but still the main result of that paper is that lipids made of fatty acids seem to be thermally unstable. The question I had was, if we go to the next slide, why exactly this happens? It wasn't explained, it wasn't explored in that paper, and I was just curious about why this would happen. Now, unfortunately, I just realized that I've converted these slides into PDF, so the next slide was supposed to be a video. But what I can tell you about this video, that anyway you can find if you want to look at the paper that it's written in the corner. What you would look at in this video are simply fatty acid-based membranes heated up to 95 degrees and then cooled down to 25 degrees. So luckily enough, I had saved some screenshots of this video. So if we go to the next slide, basically this is what you would observe in the video. You can see that at 25 degrees, you have fatty acid-based vesicles. MA stands for myristoylic acid. You increase the temperature, you start to see some weird structures, some elongated structures, up to a point in which at 95 degrees, you observe giant lipid droplets. So we go from lipid vesicles to lipid droplets just by changing the temperature. And in the slide that you are seeing now, you can see what happens when instead you cool the system down. So when you cool the system down, the lipid droplets becomes very unstable. And what you can see is that it starts to bud new membranes from the surface. So you heat the solution, you get lipid droplets, you cool the solution, you get back to lipid membranes. And the next slide shows just exactly this process, the reformation of lipid membranes. And you can see from this sort of onion-like lipid droplet, you can see new liposomes, new lipid membranes formed and detaching from that. So again, if we go to the next slide, basically these are two colored images of exactly what I've just shown you. And the question was why? Why exactly this happens? So why are we observing this change in the phase of the fatty acid in solution? And so the question came from relatively older results and papers. So if you go to the next slide, you can see that this is a ternary diagram, phase diagram for fatty acids. It is quite known that when you change the pH of a solution containing fatty acids, you can transition from lipid droplets, lipid membranes, and then lipid micelles. So basically what we were observing was a conversion that is normally due to a change in pH, but we were observing it with a change in temperature instead of pH. So again, we were interested in understanding why this was happening. And we decided to look at what was happening to the pH of our buffers that we were using to make these liposomes in the absence of the liposomes where they were heated. And so in the next slide, what you can see is that the pH of the buffer that we were using, that is the purple one, would go from 8.5 to about 6, going from 20 degrees to 95 degrees. So the pH of the solution that we were using to make our membranes was decreasing quite dramatically. While in other cases with other buffers, for example, with PBS or with phosphate, this doesn't happen. So the explanation was exactly what you can see in this graph. It is true that we are observing a transition due to temperature rather than pH, but what's really happening is that temperature is making the pH change. And so the transition that we observed from lipid membranes to lipid droplets was due to a change in pH rather than a change in temperature per se. So what we decided to do, and you can see in the next slide, was changing different buffers. What you are looking at here are absorbance profiles. Basically, if you have a relatively, if you have a solution of lipid membranes, you have a much lower absorbance value compared to a solution of lipid droplets. And so we could observe the change of the absorbance at different temperatures and understand when the transition was happening. And you can see that playing with different buffers, we would observe this transition from lipid membranes to lipid droplets at different temperatures. And so the take-home message from this, if you look at the next slide, is that fatty acid membranes are not actually thermally unstable as we assumed, as we knew. They can be thermally stable. It's just a matter of choosing the right buffer that doesn't change the pH. The next slide can show you that we've also tried to tune this temperature at which this transition happens. And you can see that if we have peptides, salts, nucleotides in solution, the temperature of this transition actually is quite affected. So the question we decided to ask at this point was, okay, so the original question was, can we reshuffle the content? And we started to study the thermal stability of lipid membranes, because it was known that at high temperature, they were not as stable as one would hope. And so the content could be released and uptaken. So we decided to look into this as well. So if you look at the next slide, what we did was we took lipid membranes containing a dye, something colored, exposed them to high temperature, and then brought them back to room temperature. And what you can see here are chromatograms that show that after the thermal cycle, we have two different populations of fluorophores. Some of the fluorophore that is on the right side is outside the solution, is just in the bulk. But some of the fluorophore, some of the dye was inside lipid membranes. You can see what we could observe with microscopy in the bottom. At the very end of the thermal cycle, we still had some membranes containing the dye. Of course, it is possible that some of them survived. So the next slide shows the control experiment, where we had the dye outside, heated them, cool them down, and again we have two populations. Some of the dye is again inside, as you can see also in the bottom. So this suggested us that at high temperature, all the content of our protocells was released in solution, and then it was retaken in once the membrane were forming at room temperature. And so what we decided to do was have a bit of fun with these systems. So in the next slide, you can see that we've mixed primitive cells with different contents. And you can see that at the end of the thermal cycle, we had a mixed content. You can do the same with the components of the membrane. So in the next slide, you can see that we took primitive cells with two different composition of lipids. And again, after the thermal cycle, we've mixed the membrane. And so we also decided to, and so what we assumed, what we understood from this was that, if we go to the next slide, basically these thermal fluctuations that were basically pH changes induced due to temperature can enable the transition of lipid membranes into lipid droplets in a reversible and tunable way. Next, we could see that the release and the re-encapsulation could be possible using these thermal fluctuations. And then next, we could see that we could use this to mix and reshuffle the content in the daughter protocells that we were forming. So if you go to the next slide, these are the two short stories that I've shared with you today. And so I'll just be very brief with the next slide, trying to just to summarize what I think is one of the next challenges, or at least the one that I'd like to focus on. And if you look at the next slide, basically combining the two concepts that I've shown you today. So I've looked at functional protocells. We've made molecules that can localize to the membrane and they can do something interesting. So what I'm interested in is understanding how this could be used to favor the reshuffling of the content, the exchange of the content with other protocells or with the environment. And with this, I'd like to thank you. The next slide is a thank you slide with all the people that helped me, forged me as a scientist, and all the participants to the work that I've presented. And then in the next slide, I just have a recap, and I'd like just to say to everyone that it would be really nice to have a chance to interact more with all of you. So if you like what I do, or if you dislike what I do, I'm always happy to interact with everyone. So please get in touch and visit us in France. Thank you very much.
Thank you for the nice talk, Claudia, and for the invitation. And we plan on having future conferences, Life in the Universe conferences. So we hope to see you there the next one. We have time for maybe one or two questions. We have a question from Pierre Durand. Pierre says, we know that compartmentalization was essential for protocells. Different authors suggest the compartments were made of lipids. Others say iron casings, like the ones discussed by Bill Martin on Monday. Is there a way of knowing which scenario was more likely?
So if you ask me, of course, I would say membranes. I think that it's quite possible that the prebiotic chemistry that is being explored in the lab could be reproduced in a variety of settings. So it really depends on what's the reason why we need compartmentalization. So if, for example, we want to explore a specific chemical reaction in a compartment just because it keeps the reactants close so that they can react more efficiently, then there might be not a strong preference for one setting compared to the other. What I can say is that we know how cells look now. So we know that cells are, let's say, their main boundary of cell membranes and are made of lipids. We also know that there are compartments inside the cell that have no lipids at all, that are called condensates or quacerbates. And I know you are going to hear from Tommaso later about this. So I think it is possible that different compartments and different settings were available on early Earth. And it is possible that different chemistries took place in different settings. I would say that if we look at biology, then we have maybe an easier answer to this question.